Good morning, everybody. Today we will start a new lesson. This is the first lesson in the part nervous system for the class third year LS section in the secondary education. We will start this new lesson that is about the chapter 9, documents 1 and 2. We will mix them together in order to let us relate them together and to make chance for gaining more time. The resting potential and action potential. Before we start, what do you have to know about the basic information of the nervous system in order to understand what we have to explain in this presentation? First, the neuron. What is a neuron? A neuron is the nerve cell. The nerve cell that constitutes our nervous system. And this nerve cell is a special cell of our body. It doesn't have the same structure as all the cells have. It has a cell body that looks like the cells we know. But this cell body has many prolongations. The prolongations of the cell body makes the nerve cell elongated. It is very tall compared to the other cells. The prolongations that you observe here that are generally short prolongations are called dendrites. And this prolongation that is generally, that is always found once in the neuron is called axon. So the neuron has generally, and we will see it further, has many dendrites. Perhaps it will have one, but it always has one axon. And each axon has at its end some ramifications that end by terminals. This is the neuron that is the basic, that is the unit of the nervous system. And what we have to learn about the neuron, that is the neuron is the essential unit that is responsible for the function of our nervous system. The nerve messages, the treatment of information, the decisions that we are taking in our brains always, our sensory messages, our motor messages that are going to make our reactions, all of them based on this neuron. We have to say before we go further that our nervous system actually contains other than neurons. It contains nutritional cells, it contains some supportive cells, but what we have to focus on are the neurons, because the neurons are only the cells related to the messages, to the nerve messages that are transmitted from one side to other in our body. Other cells may have some effects on the nervous messages, but they don't transmit messages as we will see in this presentation. Another idea that we have to learn about is about the types of neurons. Since we will not take into our minds, in our minds, that we have only this type of neurons that I show you before. We have many types of neurons, and I want precisely to focus on what we call the nerve fiber. What is a nerve fiber? Actually, you can see in this slide many types of neurons. Unipolar, bipolar, pseudo-unipolar, multipolar, but all of them have what we see here, have some long prolongations, some tall prolongations. The cell of these cells may contain one long prolongation and may contain two long prolongations, as you see here and here. And here you have only one. The thing that I have to focus on is each of these long prolongations, each of them, whether we have one or two, each of them is called nerve fiber. And sometimes also the small prolongations that might transmit messages are called nerve fibers. The thing is, this prolongation, it is called a nerve fiber, and it has a very important role in transmitting messages. Since when we have to understand the activity of the nervous system, one of the most important things to understand is the transmission of the nerve message that is done in this nerve fiber. So the nerve fiber is this long prolongation that we observe in many types of neurons and in many sites. Where do we find nerve fibers in the body? This is a very important question. We observe nerve fibers in the body, for example, in the nerve. Since the nerve is made up of many nerve fibers, here you can observe points. These points constitute the ending of the nerve fibers, and when we cut the nerve, we observe them as we observe them in a wire, in an electric wire. There are some fibers in it that we can observe, and when we cut it, we observe the endings as points. These are the nerve fibers because the nerve center, our brain or our spinal cord, that are main parts of the nervous system, and you have learned about them in the first secondary year of your education. The nerve center doesn't send each nerve fiber alone to its organ or to its effector organ or to its sensory organ as each type needs. Now, the nerve fibers are grouped together into a bundle, and this bundle, together with other bundles, are grouped into a nerve. This is the nerve, and this is an electron microscopy for a nerve where you can observe the fibers, and each of them ends in this small circle. This is the cross-section of the nerve that is very important to our presentation and to our explanation that we have to do further. This is the nerve, and this is the site where we can observe in our body only nerve fibers. We can also observe nerve fibers inside our brains, inside our nerve centers, but there the nerve fibers are either together, they are only fibers together, but they can be observed also in some parts of the nerve centers with nerve cells, with nerve cell bodies. The point here that you have to focus on is, where do we find the nerve fibers? The nerve fiber in the body can be observed in a nerve. The nerve can transmit messages to my hands, from my hands to my brain. These long structures that can transmit messages from all the parts of the body to the nerve centers, or from the nerve center to the parts of the body, are made up of fibers, are made up of long prolongations of the neurons that are called nerve fibers. And this nerve fiber is our subject where we have to study the transmission of the nerve message. How can we use an oscilloscope to measure the potential difference? This is a very important point that has to be understood. Let's see. This is an oscilloscope. On an oscilloscope, you can see a vertical axis and a horizontal axis. In our work, we will use the vertical axis to measure the volts, to measure the potential difference between 2 points. And always, in many of our studies, either in physics or in biology, the horizontal axis of the oscilloscopes measure the time units, measures the variation of time. So here you have potential difference and here you have time. And this oscilloscope measures the variation or records the variation of the potential difference according to time. Pay attention to this idea. This is a battery. We can easily connect the oscilloscope. This red wire is the positive electrode of the oscilloscope, and this is the negative electrode of the oscilloscope. If we connect them, the positive on the positive pole of the battery and the negative on the negative pole of the battery, you might observe, as you see here, this is the measurement obtained by the oscilloscope. Why do we obtain a line? Since the voltage that we are measuring is a DC voltage, is a continuous voltage. And its value can be measured by multiplying Y, which is here 3, by the scale, how much each division corresponds to. In this case, each division corresponds to 3 volts, and we have in this battery a measurement of 3 volts that is obtained by this line. What do we obtain if we switch the connections of the oscilloscope? If we switch the connections of the oscilloscope, you might observe that this line that is obtained at the beginning on the positive side of the oscilloscope, it will be obtained here on the negative side, but it will measure the same value, which is 9 volts, but it will be obtained on the negative side since the positive electrode of the oscilloscope is put on the negative side of the battery, and the negative electrode of the oscilloscope is put on the positive side of the battery. And this opposite connection will give you a negative measurement of the on the oscilloscope. Now we can start, after we taking what do we have to know before we start, now we can start by the resting potential. First, neuron and nerve message. Let's see. This slide shows you 2 neurons. One of them is myelinated. We will study the myelin further and deeply in the next slides, in the next lessons. And this is a non-myelinated neuron. The myelin is something that covers the axon. The neurons, as we said, can transmit nerve messages. They transmit nerve messages inside our body. This transmission allows me to see when the neurons transmit messages from my eye to my brain. They can make us move when they transmit some messages from our brains to our muscles. These messages can be transmitted from both types of neurons, but now you will observe this transmission will be much slower in the non-myelinated fiber. These transmissions make us pose some questions. What is the nature of this message? What is the nature of this nervous message? Do you have some hypothesis? Try to think. And you have the time, if you have the time, to pause this presentation and think about the nature of the nervous message. Please do it. Since when you think, you might have more chance to understand what I will explain further in this presentation. Now, is the nervous message electrical? Is it like the electricity that passes inside the electric wire? We should study it in order to find its nature. Now, how can we study it? This is a nerve fiber, and this is the myelin that covers it. And this is the nerve fiber. It is inside the myelin. And we can measure the potential difference of a nerve fiber by using what? An oscilloscope. This is an oscilloscope. This is actually a voltmeter, and this is the screen of an oscilloscope. We can observe here when we put one of the electrodes, that is the positive electrode of the oscilloscope, and we put it where? Inside the fiber. This is the negative electrode of the voltmeter or of the oscilloscope, and we put it outside the fiber. You can observe that when we put, at the beginning here, when we put the oscilloscope inside, we observe a decrease, we observe a negative potential inside the fiber. And when we take it off, the potential returns to zero. So this potential that we obtain, this potential difference that is equal to minus 70 millivolt, is the potential that we obtained between inside and outside the fiber. This is the value of what we call, this potential is called what? Can we call it a nerve message? You have to think. Please pay attention and think about what can we call this potential difference that we observe inside a nerve fiber. Can we say in this potential difference that is equal to minus 70 millivolt and that is constant as we see here, can we say it is a nerve message? Do you have any answers? The answer is no. This is not a nerve message. Why? Why that the obtained potential difference can't be called nerve message? Actually, since this is an isolated fiber that does not transmit any nerve message. There are no nerve messages inside this fiber since it is isolated. It is isolated from the body and it can't contain any nerve message since it is not in activity. And since this fiber is at rest, this potential will be called resting potential. This is the title of our first part of this lesson. How can a nerve fiber at rest, as we see here, how can it have a potential difference across its membrane? This fiber is at rest. What makes it having this potential difference? We will see here in this presentation a cross-section of the fiber. The fiber that is elongated appears as a circle when we cut it and when we make a cross-section in it. This is the cross-section of the fiber, and this is here the cell membrane. And we can observe some things inside and outside it. What are these? First, this small red point represents the Na+ ions. This blue point represents the K plus ions. As you see here, you can observe that outside the nerve fiber, there are big number of Na plus ions. And inside of it, there are big number of K plus ions. It doesn't mean that the Na plus ions do not exist inside. Inside the fiber, there are some tiny concentrations of Na plus ions. And in and outside, there are some other tiny concentration of K plus ions. But the big amount of K plus is found inside, and the big amount of Na plus is found outside. These represent the proteins that are negative in their charges and that might compensate some of the positive charges that we obtain inside. And we have also here some points, the green points, that represent other negative charges, and they might be Cl minus, per example. Now, you can see the composition of, or the structure of the cell membrane. This cell membrane, as you see here, contains a big number, as we see, of Na plus ions outside and some of K plus. This figure will make you sure about the presence of K plus outside. And inside you have the opposite, completely opposite thing, big number of Na plus ions and K plus ions, sorry, and some Na plus ions. So we have a big number of K plus ions that are green, as we see here, and very small number or concentration of Na plus ions, as you see here. And the other ions that I put in this figure are not shown in this one. There are some proteins that are negatively charged and other negative charged ions also inside. And we have some negative charges outside the fiber. But this representation doesn't explain to you what is the origin of this potential difference. If I let it as I see here, I will not observe any potential difference, or I can't understand this potential difference, actually. Now, if we let this fiber as it is, you have to see, as you see here, that the K plus ions will move outside. Why? Observe here, you have these green channels that we call leak channels. These channels are some protein channels that are found in the cell membrane and that can make the K plus ions diffuse. And since K plus ions are found inside more than outside, these ions have to pass from the high concentration medium, the medium of high concentration, to the medium of low concentration. And this diffusion is the cause of the passage of the K plus ions to the outside. This is how we can observe outside the figure, outside the fiber, some K plus ions. And another thing, this is what we call a diffusion according to the chemical gradient of K plus. Chemical gradient means that the ions diffuse from the medium of high concentration to the medium of low concentration of this ion. You can now ask yourselves, do the Na plus ions also diffuse in this model? You have to observe carefully. Do you have here some Na plus channels? Observe it well. Take your time to observe that you have here Na plus channels. And the Na plus, as you see here, is going through the channel to the inside of the fiber. Correct? Let me see. This is how Na plus passes to the inside. But why Na plus passes? Na plus passes according also to the chemical gradient, since its concentration outside the fiber is higher than its concentration were inside the fiber. But you can also say that this might have other causes that we will study further. Now, the concentration will make Na plus pass from outside to the inside. let's count. 3 Na plus ions pass to the inside, and 10 K plus ions pass to the outside. Why? Look at here another time and try to find an answer for this question. Why only a few amount of Na plus ions pass to the inside and a big amount of K plus ions pass to the outside, or bigger amount? Why? Try to think about this idea, about this question, by observing this figure. And if you can't pause the video, please do it and think. Think carefully, since this type of reflection is what led you to remember again these information when you have to. Observe here. I have here more number of K plus channels than Na plus channels. So the big number of K plus channels make the cell membrane highly permeable for K plus, and it makes it very slightly permeable for Na plus. This is why a big number of K plus ions can cross the membrane toward the outside, and a small number of Na plus ions can cross the membrane to the inside. What do we obtain if this diffusion happens? We will obtain more positive ions outside. Correct? Think about it. If you count the positive charges outside, you will find them more than the positive charges inside. And this is that cause a negative charge that appear inside the fiber and a positive charge that appear outside the fiber. This will give you a potential difference between inside and outside the fiber. And as we saw, this potential is the resting potential that is found in a nerve fiber at what state? At the resting state, not at the state of transmission of nerve message. This is why when we connect an oscilloscope, the positive electrode inside the fiber and a negative electrode outside the fiber, you will see a negative potential difference. It is negative since the positive electrode is put on the negative side and the negative electrode is put on the positive side. This is why it is negative. And it is equal to minus 70 millivolt to show you the difference between the concentration, between the whole number of positive ions that are inside and the whole number of positive ions that are outside. This is the resting potential. It is equal to minus 70 millivolt. And this number is not a precise number for all fibers. It might differ from one fiber to another according to the species, according to the state of the fiber, according to some physiological states of the nervous system. So in this animation, you will observe the same ideas again in order to understand some of them. The animation will take you in another way to understand this information. You can observe here the cell membrane, and these are the channels as you see. You can easily remember that the big number of channels are the K plus channels, and the small number of channels are Na plus channels. This is the potential difference that we can observe across the membrane, since it is charged positively from outside and negatively from inside. Why? Since as we saw, and like we see here in this animation, Na plus ions will cross the membrane to the inside, but more K plus ions are crossing the membrane to the outside. According to the concentration gradient, K plus go outside, its concentration outside is lower, and Na plus goes to the inside. Inside has less concentration of Na plus ions, And this diffusion that is not equal across the membrane for K plus and A plus ions make the outside positive, because the whole number of positive charges are now outside, more than the whole number of positive charges that are inside. And this is what makes the charge outside the membrane positive and inside negative. This number, minus 90 millivolt, will be obtained if Na plus does not diffuse inside. But with the presence of diffusion of Na plus inside, the potential will be minus 70 millivolt. And this pump is called Na plus K plus pump. This pump consumes energy in the form of ATP, and it prevents the balance of ions from between inside and outside. Since if Na plus ions are balanced between inside and outside the membrane, and K plus are also balanced between inside and outside, we will obtain an electrical neutrality, and the potential difference between the intracellular medium and the extracellular medium of the membrane will be null. The pump of Na plus K plus, as we saw, it pumps K plus inside to let always the K plus ions inside high to continue their exit, more than the entry of Na plus that is maintained high outside the membrane according to the pumping of the Na plus ions by the pump from inside to outside. So this pump consumes energy. It is called Na plus K plus pump. It consumes energy in the form of ATP in order to pump K plus inside and Na plus outside, in order to maintain this difference in ionic concentrations across the cell membrane, to maintain always the potential difference equal to minus 70 millivolt, as we explained. And this is what we called resting potential. So as a general conclusion about this problem about how can a nerve fiber have a resting potential and have a potential difference when it is at rest. The potential difference origin, and it is what we called resting potential, is the selective differential permeability of the cell membrane. Selective differential means that the cell membrane selects Na plus and K plus to move them across, and differential since there is a difference between the amount of K plus that is moving across the membrane and the amount of Na plus that is moving across the membrane. This difference in the permeability of the cell membrane leads to a difference in the concentration of ions between inside and outside. Here I mean the concentration of all positive ions and the concentration of all positive ions inside and outside is the origin of a difference, as we saw, in the potential between inside and outside. And this difference of ions concentration is maintained by the action of an Na plus K plus pump that consumes energy, and as we saw, it pumps Na plus always outside and it pumps K plus always inside the fiber. And this is the origin of the resting potential. This resting potential is very important for us in order to understand the next idea, the idea of action potential, and that I will explain. I will make a presentation about the action potential in the next lesson. So be with us in order to understand the relation, as we said, between the resting potential and the action potential, and how the resting potential is important, is crucial for the obtention of an action potential that will be studied further. Thank you.